Good afternoon. I want to thank you for joining us for our MDICx webinar on surrogate sample draft framework. My name is Carolyn Hiller. I'm the Program Director for Clinical Diagnostics here at MDIC. For those of you who are not familiar with MDIC, the Medical Device Innovation Consortium is a nonprofit public-private partnership dedicated to advancing the regulatory science of medical devices. We create tools and methods to help bring technology to patients faster, safer, and more cost-effectively. Now, I'd like to introduce our speakers today. Our speakers include Dr. Suzanne Schwartz. She's the Associate Director for Science and Strategic Partnerships in the Office of the Center Director for CDRH in FDA. We also have Dr. Alberto Gutierrez. He's the Director of In Vitro Diagnostics and Radiological Health in CDRH, also at FDA. And we have April Viokas. She's a Director of Regulatory Affairs at Abbott, and at MDIC, she is our Surrogate Sample Working Group Chair. We'll reserve the last 10 minutes for Q&A. You can enter your question in the chat box, and my colleague Stephanie Christopher will read the questions. Now, I'd like to turn the webinar over to Dr. Schwartz.
Thank you very much, Carolyn. It's really a pleasure to be here. First time for me on the MDICx series webinar, hopefully the first of many going forward. Stephanie, if you could turn to that introductory slide, the slide titled Value of Collaborations to Advance Regulatory Science. And just, again, perspective in terms of where I'm coming from within the Center for Devices and Radiological Health, leading the programmatic portfolio that encompasses science and strategic partnerships. What does that actually mean? As we work towards developing or helping cultivate the development of these different types of tools and approaches that are necessary for the FDA in order to inform decision-making, regulatory decision-making, our program is involved in helping shepherd that process. So if we turn to the next slide, what is regulatory science? And I think that everybody would agree that the necessity for as much transparency, consistency, reproducibility, and predictability with regard to decision-making for medical products and medical devices specifically is really important. And our decision-making from on a regulatory framework needs to really be grounded in evidence and in science. So how do we get to that body of regulatory science, and what does that actually translate to with respect to the outcomes and the benefits? Once more, you know, it is about developing these kinds of methodologies, tools, standards that is going to enable that type of consistency with respect to assessing or evaluating the performance and the safety and effectiveness of devices. Why is it important to do so, or what are the benefits? Well, we believe that by virtue of setting up this type of methodology and body of science, that is going to enable products to get to market a lot faster with a safer profile and in a more cost-effective manner as well. Next slide, please. Now, regulatory science is not restricted to any one particular area within the entire life cycle of development. In fact, it really spans across the entire life cycle, as is depicted here on this particular slide, from the moment of discovery and ideation, really all the way out through a product that has already been put into distribution, where monitoring on the post-market side becomes important. Next slide, please. The regulatory science encompasses really multiple domains with regard to how do we get there. What does that mean for us at CDRH? Number one, it's the research itself, and so the center supports the different regulatory and scientific needs in order to inform that decision making. We'll do that by leveraging expertise as well, through the development of standards, working with standards organizations, and very importantly, through public-private partnerships that allow us to further expand upon existing regulatory science. And that really brings us to the next slide, MDIC. Why is it important? MDIC, as Carolyn mentioned at the forefront, is a public, not-for-profit, public-private partnership. And this concept of being able to drive towards getting safer products out to the patients faster and in a more cost-effective manner means that we really need to be able to align, accelerate in order to achieve those particular goals. Next slide. And how does MDIC really tackle or address these challenges? What are the strategies? Through several multi-pronged approaches. Number one, creating that forum for collaboration and dialogue. Secondly, by making strategic investments in regulatory science. And third, through the provision of tools that will enable or drive innovation forward. Next slide. We all believe that collaboration is absolutely critical when it comes to solving some very, very difficult problems and difficult challenges. And regulatory science is indeed big science. So this concept of really trying to pool the resources that we have in the ecosystem at large, pool the subject matter experts, the novel ideas, is really all critical in terms of being the ingredients towards bringing about new products, new understanding of regulatory science, and that's developed through the emphasis on collaboration. MDIC promotes these collaborations, and it really brings together the entire ecosystem on the public-private side, and to include manufacturers or industry, government partners, as well as other stakeholders. Next slide. It really brings us full circle to what MDIC and regulatory science has to offer. What is that value proposition towards our unified mission of getting patients access to safer products, new technologies in a manner that's more expeditious and more cost-effective. So with that, I want to just thank everybody for the opportunity to do a little bit of framing around MDIC and regulatory science as we work together, FDA, with this really important collaboration. And I'm going to turn this back to, let's see, Alberto.
Yes, thank you, Dan. Next slide. So I'm just going to address specifically the interactions that my office, or part of my office, is having with MDIC around really the diagnostic, or more closely, in vitro diagnostic issues. So next slide. So just like our collaboration overall in the MDIC program, really the goal here is to find projects that will improve diagnostic product development and testing based on the application of novel regulatory science approaches, and to do this with the goal of speeding patient access to safer, more effective diagnostic technologies. Next slide. to do so, we started really with 4 areas that we wanted to tackle early on. And these are 4 different areas in the total product life cycle, if you like. We wanted to have a better understanding of what clinical evidence was needed when, to see if there were better or if we could define better what kind of uses we can have for contrived sample, when is it appropriate, and what kind of uses one can have with contrived or surrogate samples, to figure out how to better have interoperability in the health records or electronic health records, and lastly, what's behind, if you like, incremental improvements and what needs to be done to validate those, or how do companies approach those things. And in general, these were the 4 topics that we chose. The one that has really taken off, and which April is going to be presenting today, is what we have done with contrived and surrogate sample use. Next slide. So that group actually has come a long way. It started with a survey that went out to industry and other parties. FDA also participated in the survey. And on March 23rd, we had a webinar in which we presented the results of the survey. Today, we're issuing the draft framework, and the seminar April is going to be presenting the highlights of the draft framework. And then there's going to be a comment period that will be open for those interested to comment on the draft framework. Based on those comments, we will edit the framework, and we are hoping to finalize the framework around October of this year. So without further ado, next slide, I will let April present the draft framework. April?
Thank you, Alberto. I'm very excited to have this opportunity today to announce the release of the draft surrogate sample framework and provide to you an overview of key elements of the framework. Next slide, please. As just mentioned by Alberto, here are a repeat of the key dates, along with a link to the MDIC web address where the draft framework is located, as well as on that same link, access to a comment form where you can submit your comments regarding the draft framework. The draft framework is available for comment beginning today, and that comment period will run through the end of June, June 30th. Once the comments have been received, the work group will take those comments, review them, and use them in the process to finalize the framework, which is noted is scheduled for publication later this year. Next slide, please. Before reviewing the framework, I'd like to take a few minutes to really recognize the members of this work group. As you can see, we have members from industry, from FDA, as well as expert advisors. And the draft framework and the content that will be reviewed today during the webinar are the results of ongoing collaborations among the individuals of this work group. The individuals have spent quite a bit of time and effort in getting to this point, and I wanted to take this opportunity to express my appreciation and gratitude to the team for all the effort put into this particular project. Next slide, please. So IVD stakeholders recognized that when there's difficulty obtaining clinical specimens, this can delay the development of innovation and improve diagnostics, ultimately impacting patient access to these products. To address this issue, the MDIC Surrogate Sample Work Group has been charged to establish a foundation for the use of surrogate samples to support IVD product validations and the regulatory submission process. having a clear and predictable foundation fits squarely within the goals of MDIC of speeding patient access to safe and more cost-effective diagnostic technologies. Next slide, please. As noted, the work group conducted a survey, and we've done research into historical practice. And this has demonstrated that there are a wide variety of reasons for using surrogate samples as part of diagnostic development and validations. Surrogate samples provide an option when naturally occurring specimens of biological origin are rare or difficult to acquire. An example of this would be with a quantitative test where there are challenges acquiring a sufficient number of specimens across the measurement interval of the test. And this may be true especially at the high end, low end, or at the medical decision point. This is an example of where surrogate samples can be used to help address these challenges and move forward development of such products. Ethical considerations is another reason why one may turn to the use of surrogate samples. Examples may be invasive sampling methods or concerns about drawing large volumes of clinical specimens from sick patients. And in these situations, there is a role for the use of surrogate samples. Surrogate samples may also help simplify sample acquisition and characterization activities. And for certain types of studies, the study demands, the scientific demands, may require the use of specialized or prepared samples to meet different criteria, such as the need to have a large volume of sample or the need to address situations which are difficult to find in the general population. Next slide, please. Before delving into the details of the framework, I wanted to spend a few minutes describing the work group's approach to the framework. A little bit about what the framework is and then what the framework is not. First, one of our objectives is to establish a common language to facilitate and guide discussion in this area. Surrogate, contrived, altered, pooled, and spiked are just a few examples of common terms that are used in this space, and therefore aligning on definitions and terminology has been a key element and focus of the framework. Next, as a work group, we looked at striking the right balance between providing general information to guide decision processes related to the use of surrogate samples, while also recognizing that there are exceptions or alternate considerations that may exist for particular types of studies, and have designed the framework in a way to balance these two somewhat competing aspects. Although the framework includes information by study type, for example, there's a section on linearity or in a section on precision, it is not intended to provide all the elements of study design that one should consider. This information on study design exists in current formats such as standards and guidelines, and we really focused and designed the framework to emphasize the use of surrogate samples in these situations and intentionally steered away from general study design. Lastly, we centered on a hierarchical approach to guide the selection of the appropriate types of surrogate samples. Given the breadth of analytes, technologies, and other factors, this hierarchy is intended to serve as a working foundation which can be applied more broader and in a more general context to in vitro diagnostics. As you review the draft framework, we look forward to receiving your feedback aligned within these objectives. Next slide, please. So as noted in the draft framework, we aligned on the term surrogate sample and the following definition. A surrogate sample means material used as a substitute for body fluid or tissue taken for examination from a patient. We conducted a search of relevant standards such as ISO standards and CLSI standards, as well as FDA guidance to locate a definition. When we were unable to locate an existing definition, we developed this definition based on collective experience and accepted definitions of related terms, such as biospecimen and surrogate material, which can be found in existing standards. Initially, when we approached the topic, and some of our work with the survey will reflect this, we used the term contrived interchangeably with the word surrogate. But as the team progressed and based on additional research and common dictionary definitions, we decided that a contrived sample is an example of a surrogate sample, but that the more comprehensive term surrogate best describes the types of sample within the space. And therefore, in the draft framework, you will see that we have adopted the terminology surrogate sample, and the definition applies to that term. To increase the clarity of the definition, we've included the following examples, which are materials supplemented with an analyte of interest, pooled specimens of biological origin, material created to have functional characteristics of a body fluid or a tissue, and material comprised of a combination of an analyte that emulates the analyte of interest and a material created to have functional characteristics of a body fluid or tissue. The addition of this fourth example occurred later in our process, and it is intended to recognize that surrogate samples may not only contain surrogate matrices, but may also include surrogate analytes. Next slide, please. Within this definition of surrogate sample, we further describe samples as falling into one of three categories. The first is supplemented, which means individual specimens of biological origin spiked with an analyte of interest. The second is pooled, combined individual specimens of biological origin that may or may not be spiked with an analyte. And then the third is simulated, which means treated materials of biological origin, or it also means artificial materials created to have functional characteristics of a body fluid or tissue that may or may not be spiked with an analyte of interest. Next slide, please. The framework contains a surrogate sample hierarchy that is structured around these three categories: supplemented, pooled, and simulated, and accounts for both matrix and analyte. This hierarchy has its origins in the CLSI guideline EP06-A, Evaluation of Linearity, which describes a hierarchy for the preparation of samples used to complete linearity studies. We have expanded on this concept in the framework to create a general hierarchy or approach which can be tailored by study type and individual situations. The hierarchy not only stratifies surrogate samples across the three main categories, supplemented, pooled, and simulated, but also within each category. So as you move down within a particular category type, you will see a priority or a preference listing of selection of surrogate samples. For example, if acquisition of a matrix through individual specimens or pooled specimens is not feasible, so if category 1 supplemented and category 2 pool are just not feasible options for the particular situation, one would then next move to the box indicated as simulated. However, even within this category, the different types of simulated surrogate samples are arranged in a hierarchical format, beginning here with treated patient sample pool and ending with organic solvents. Additionally, the hierarchy considers analyte in addition to matrix and whether or not the analyte is artificial, that is, it emulates an analyte of interest, or is naturally occurring. Next slide, please. After identifying the reason for using surrogate samples for a particular study type, the framework sets forth general principles to assess and help identify the factors that are most pertinent to the study in which the surrogate sample is being considered for use. One would first identify these factors before using the hierarchy described on the previous slide. So first, in terms of these general principles, is to clarify the objective. Are the surrogate samples intended to supplement naturally occurring specimens in the study, or are they intended as a replacement for naturally occurring specimens? What are the critical factors related to the performance study itself? For example, is preserving biological variability a key element of conducting the performance study? Or might the performance study be one which requires large volumes of sample in order to complete it in a controlled manner, and therefore meeting scientific demands of the study is what is triggering the use of a surrogate sample. In other situations, one of the key factors may be the introduction of a variable that can only be studied in a surrogate setting to understand how the test performs under stipulated conditions. In addition to considering factors of the study, it's important to consider whether or not the test is a qualitative or quantitative test and use this information in the consideration of the type of surrogate sample that will be used. Also important is to consider the effect of extraction method, if applicable, and what its impact is in increasing or decreasing biological variability, and the role this has in the preparation of the surrogate sample. Next slide, please. When selecting the surrogate sample matrix, consider which functional characteristics of the naturally occurring specimen are most relevant for the study type. So here, one would consider whether there are aspects of the surrogate sample that would interfere or otherwise influence test performance in a way that is inconsistent with naturally occurring biological specimens. When a matrix is artificially prepared, describe the characteristics of the naturally occurring specimen that are represented or mimicked by the surrogate sample. Here, one would consider factors to minimize differences between naturally occurring specimens and surrogates, and this would include items such as viscosity or endogenous cellular components. The next general principle to consider is the use of established references and methodologies. The team recognized that in some areas there are existing standards or guidelines regarding the use of surrogate samples for specific study types, as well as FDA guidance that exists related to particular analytes or technology. And as part of the general principles, we wanted to bring forth the recognition of what exists today and include that as a factor to consider when approaching this topic. After identifying the relevant factors and the reason for using surrogate samples, one would then use the hierarchy to identify a surrogate sample that is similar to the specimens that will be used in the clinical setting, as well as also meeting the demands of the study type. Next slide, please. And although this framework begins with a general approach, we've also included more tailored and study-specific information for the study types listed here on this slide. Well, the time necessary to discuss all the elements that are contained in the framework for each of these study types is beyond the time allotted for this webinar. The next set of slides will address each study type by focusing on key principles and considerations that are representative of the type of information contained within the framework to give you an idea of how we've approached this balance of general information, and then noting that there are unique items that may need to be considered within particular types of studies. Next slide, please. By way of illustration, the framework acknowledges that for linearity studies, surrogate samples are widely used. However, it also provides individuals with considerations such as that for these types of studies, the preservation of biological variability is an important factor when conducting linearity studies. And thus, this factor would weigh into the selection of the appropriate surrogate type when one is looking at the hierarchy. Additionally, as an example, this is an area in which there is existing CLSI guidelines, and this is noted here on the slide, but it is also contained in the framework as well. Next slide, please. For analytical specificity, the framework describes the situations in which surrogate samples are more frequently used, which is when clinical specimens are unavailable or it's not possible to obtain specimens with known amounts of potentially interfering material. Additionally, this slide demonstrates the incorporation of the hierarchy into the process by identifying the category supplemented as the place to start when considering surrogate samples for analytical specificity. Next slide, please. The information for precision reproducibility studies is another example recognizing the use of surrogate samples to conduct studies, but it also illustrates how two study factors weigh into the type of surrogate sample used. And this would be acquiring sufficient sample volume to conduct the study, but also maintaining sample variability. So one would take these two factors into consideration as the hierarchy is reviewed and use this information to help select the appropriate surrogate sample type for conducting these studies. Next slide, please. For detection capability studies, the slide demonstrates a key factor for consideration is the sample matrix itself. And this is in addition to the study factor of preserving biological variability. Incorporation of use of the hierarchy and why one may need to move from the first main category on the chart, supplemented, to the second main category, pooled, is explained here. So in terms of the hierarchy itself, one would begin at supplemented as a means to preserve biological variability. However, this may not meet the demands of the particular study, and therefore it may be necessary to move to the next category, pooled. Well, when pooling specimens, there are different ways to approach that, and this is demonstrating and providing additional guidance on how might one prepare the samples, the pooling of the samples, in order to best meet the needs of the study. Next slide, please. Matrix comparison illustrates specific situations in which one would consider the use of surrogate samples. For example, covering the measuring interval of the test, obtaining patients with primary sample values close to the medical decision level. There may not be enough patients with positive test results to complete the comparison, or there may be difficulty acquiring a sufficient number of samples close to the cutoff. And it is in these situations where one would consider the use of surrogate samples as part of the matrix comparison study. In addition to describing important factors to consider when preparing surrogates under considerations, this also takes into account that the way in which one prepares a sample may introduce confounding factors into the study, such as how one dilutes and adds additional material to the surrogate. And therefore, the guidance provided here is designed to address this preparation aspect to minimize the introduction of confounding factors into the study. It's also an illustration of an area where there is not existing guidelines at this particular point in time. And so therefore the context of the framework is really the primary type of information that is available at this point in time to guide the use of the hierarchy for this study type. Next slide, please. The method comparison study provides an example of when the use of surrogate samples is generally more to supplement rather than to replace naturally occurring specimens. And noting here in particular that preserving individual biological variability is an important factor for this study type. So therefore, when one is looking at the sample hierarchy, this is an important factor to keep in mind when looking at how best to prepare the surrogate sample. Additionally, this provides some guidance in terms of how might one address the need to preserve biological variability during the preparation of sample for this particular study type. So in regards to pooling, pooling is not recommended as the first approach. However, there may be other reasons to do pooling in order to conduct the study, and that in that time, the use of the sample only once in this pooled setting is recommended to preserve variability. Next slide, please. For specimen stability studies, surrogate samples may be appropriate under defined conditions, such as when one is unable to acquire sufficient sample volume, or targeted analyte levels, such as those at the medical decision level, are difficult to acquire. Here it's noted, though, when one is looking at the use of surrogates for conducting specimen stability studies, there are important factors which are a matrix resembling the naturally occurring specimen matrix, and that consideration of the extraction method that may be applied as part of the test procedure is considered as one is selecting the surrogate sample. Next slide, please. Similar to other areas where the use of surrogate samples is generally recognized, the reagent stability information acknowledges this point, and it also illustrates the use of the hierarchy and the stratification process contained within the hierarchy as a means to select the appropriate type of surrogate. So here, for example, one would begin with the category of pooled to select the sample type. And within the category of pooled, there is further stratification. And one would look at this to ensure that the selection of the sample is in line with what is most representative of a clinical specimen. Next slide, please. For the instrument carryover study, this slide is intended to illustrate that there are key elements of the specimen matrix that is an important factor to consider when identifying an appropriate surrogate type. And then here, there are additional examples that have been added, such as excessively mucoidal specimens, hemolyzed blood samples. By providing these examples, it's intended to supplement and further emphasize why there is this need to consider the matrix and its relevance to the conduct of these types of studies. Next slide, please. For clinical performance evaluations, there is less existing reference material and data on the use of surrogate samples. And you can see this reflected within the framework by its focus on the use of surrogates in this area to supplement rather than replace naturally occurring specimens, and that there is, in this situation, emphasis on the use of well-characterized surrogate samples because of the type of study we are talking about here. Now, these last 10 slides were intended to demonstrate how elements of the general approach, the sample hierarchy, as well as specific information related to the study are applied and tailored for each type of study. And although limited elements were touched upon for this presentation, more detailed application is contained in the draft framework, again on this interplay between the consideration of general principles, but acknowledging the tailoring to the specific study type. Next slide, please. So in terms of deliverables and next steps, as mentioned previously, the survey report was published in March of 2017 and again is available on the MDIC website. There is a recording available of the webinar that occurred along with the release of that report. As noted again, the draft framework is available today and can be downloaded from the MDIC web address noted on this slide. There will be a recording of this webinar as well, which can be accessed via the MDIC website. As a reminder, the public comment period on the framework begins today and runs through June 30th. We, as a team, look forward to receiving feedback on the framework and are looking forward to, again, getting the different perspectives that exist on the use of this framework and, you know, even recommendations for areas of refinement. The target date for publishing the final framework is October of this year. And with that, I'd like to go to the next slide. And use this time again to just provide to you, the team members on this project, and again express my appreciation to the team as a whole for the good collaboration, the good discussion, and all the efforts to getting us to this point in time where we are able to release the draft framework. So I will now turn the program over to Stephanie for the Q&A section.
Great, thanks, April. As Carolyn mentioned at the beginning, you can submit your questions through the chat box on the WebEx screen, and some of you have started to do so already. The first question, will the framework tell me the specific type of surrogate sample that I should be using for my assay?
Okay, thank you for the question. So the framework does not get into that level of specificity. What the framework does, though, is it is intended to provide the important points to consider and a process for evaluating these factors and applying them within the context of the hierarchy. And this was done in order to produce a deliverable that would have a broader application across the different types of IVDs, as opposed to trying to get very specific for specific types of IVDs, which we felt would not have the same impact to the stakeholder community as a broader approach.
Great, thank you. Second question, someone posted, I'm interested in learning more about the surrogate sample hierarchy. Can you walk through this chart in more detail? And I'll bring that back up for you, April.
Sure. So again, as noted, the hierarchy is presented in a format that includes both matrix and analyte. It recognizes that samples may be spiked or not spiked. The large arrow within the framework is intended to demonstrate the staggering of the boxes, and it represents a general preferential. However, it is noted that the actual preference of a surrogate sample type is situation dependent, and may vary from that indicated by the figure. So even though one would start at the top and move down the hierarchy, it is recognized that there are additional factors, as described previously, that weigh into this decision-making process. And then as one looks at the hierarchy, within each section there is this further stratification of the different types of, like for example, the different types of pooled specimens or pooled samples, and this stratification is intended to present these options to users in a manner that really shows this preferential approach.
Great, thank you. I've had a couple of people ask about whether slides or materials will be posted, and I just want to share that, as April mentioned, the video from today's webinar will be posted on the MDIC website, and certainly all of the material that's been posted here is in the framework itself, and I know the whole team, including April and Carolyn here, would love to have your feedback on that report. So I encourage you to go to MDIC's website and download that report and supply this team with your feedback. Looks like I may have one more question here. Hold on one second as I pull it up. This person has asked, I'd like to know if any of the aspects of this framework are mandatory when considering the preparation of submissions to the FDA. Is this more of a guideline rather than a binding document?
So from an MDIC perspective, again, this is a public-private partnership that's again focused on advancing regulatory science. So this is not an FDA document, although we do have very active membership from FDA, it will be released as an MDIC document, and we would hope that it would be of value to others in the space, such as, regulatory agencies and standards bodies, but it is being issued as an MDIC document, and I'm not sure if anyone from FDA would like to add anything more to that.
Yes, this is Alberto Gutierrez. No, you're right, April. This is a document issued from MDIC, and it's not an FDA guidance.
Great, thank you. Well, Carolyn, I don't see any additional questions coming in.
Okay.
Unless you want to make some closing remarks?
Sure. Again, I want to extend an extra thank you to the working group who pulled this all together, and to our speakers who joined us today. A reminder, you can download the framework at our website, that is mdic.org, and we are in the program area, Clinical Diagnostics. Please provide your comments. Encourage your colleagues to also comment on the document through the end of the month. And a recording of this webinar will be available in the near future. Thank you for joining us, and we wish all of you a good day.